All right, everyone, welcome back to episode 4 of our podcast on BIO110. Sounds like today's topics are going to focus on helping you succeed, right, Dr. Rosario?
Yep. One of the things I love about teaching is figuring out where students might struggle and then tweaking things to make it easier for them to really dig into the material. I made a couple updates this week that I think are going to help a lot with that.
Okay, let's start with the quizzes. I heard you've made some adjustments there?
Oh, yeah, absolutely. So quizzes 3, 4, and 5, they're now open. These were originally locked until closer to certain lecture dates, but now students can tackle them at their own pace. I gotta say, giving flexibility like this has been a game changer for some.
So they've got the freedom to do them earlier if they want to, or closer to the due date.
Exactly. Personally, I'd recommend doing them closer to when the material wraps up in class. It's like fresh in your head, you know? But hey, if you're someone who likes to plan ahead, go for it. What's important is using these quizzes to really reinforce what you're learning. Studies show active retrieval, what you do when quizzing, actually helps lock in new information. So, my advice? Use these as like your tool, not just an obligation.
Right, better retention and learning, not just a box to check. But I also know you've got more tools up your sleeve. Let's talk about that study guide for Lecture Exam 2.
Oh, yes, the study guide is live, and I cannot stress this enough. It's like your map to success for the next exam. It covers everything we've discussed, including today's lecture, and I think it'll be especially helpful for understanding cell structure and function. A lot of students tell me these guides are lifesavers because they can see exactly what key topics to focus on.
And it sounds like it's more than just a list of topics. It's really targeted to help them prep strategically?
Oh, totally. It's designed to guide you, not overwhelm you. Think of it like a highlighter for your notes. If you go through it systematically, you'll be in great shape for the exam. Remember, preparation is key. Don't cram. Start using the guide now and build that understanding step by step.
Solid advice. So to recap, students now have early access to quizzes 3 through 5, and the Lecture Exam 2 guide is up. Key tools for anyone looking to stay ahead.
All right, building off what we talked about earlier with those study tools, let's dive into one of the coolest concepts in biology, understanding how cells are organized. Specifically, we're going to explore what makes prokaryotic and eukaryotic cells so distinct. You ready?
Absolutely. But before diving in, quick reminder. These are the two broad categories of cell types, right?
Exactly. At the most basic level, all living organisms are built from one of these two kinds of cells. So prokaryotes, think bacteria and archaea, are like these simple, efficient, single-room spaces. Everything happens in one big open area. Meanwhile, eukaryotic cells, which make up plants, animals, fungi, and humans, are more like multitasking mansions with specific rooms.
Okay, that's a pretty vivid image. A studio apartment versus a mansion. But how do those differences actually look inside the cell?
Oh, it's wild. Let me highlight some big ones. Prokaryotic cells don't have a nucleus. Their DNA is just free floating in the cytoplasm. Eukaryotic cells, on the other hand, have this super cool structure called the nucleus. It's like the command center housing all the cell's DNA safely locked away.
So the nucleus is like the private office in that mansion analogy?
Yep. And here's the kicker. Having that private office lets eukaryotic cells organize and protect their genetic material much better. It's a game changer when it comes to complexity and specialization, but that's just the beginning. Eukaryotes also have these compartments called organelles, little rooms dedicated to specific functions. You've got ones for energy production, protein synthesis, waste management, the works. Prokaryotes? Nah, one big shared space for everything.
Got it. So prokaryotes are more simplified, streamlined even, but eukaryotic cells are essentially compartmentalized powerhouses?
Exactly. Here's an example to bring it to life. Think about a bacterial cell. Super efficient, handles everything in a compact space. But if you compare that to, say, a human cell, it's like going from a pop-up food truck to a fully loaded restaurant with a dining area, kitchen, everything.
That's a great analogy. But why do eukaryotic cells need all these rooms or organelles?
Amazing question. It comes down to specialization and efficiency. By separating tasks into different spaces, eukaryotic cells can create specific environments tailored for certain functions. For example, in one room or organelle, you might have a high energy environment for making, say, ATP. Then in the nucleus, you've got DNA storage and protection. Everything runs smoother this way. It's like cooking in a kitchen instead of, I don't know, trying to make lasagna in your living room.
Makes perfect sense. And I'm curious about something else. What does this mean in terms of life? Like, why are cells considered the smallest units of living organisms?
Ah, yes. You're talking about the famous cells are the basic units of life idea. Okay, here's why. A single cell, even if it's just one bacteria, can carry out all the functions we associate with life, sensing the environment, obtaining nutrients, reproduction. But go any smaller than that, like down to an organelle or DNA strand, and you lose that independence to function on its own.
So the cell is like the smallest complete system that can still survive and operate independently?
Exactly, exactly. It's like a self-sustaining city, right? You've got energy plants, factories producing proteins, waste disposal systems. It's all there. And what's wild is all of this is happening at such a microscopic scale you can barely imagine it. Cells are utterly fascinating.
No kidding. And what's amazing to me is how cells, no matter the type, still have some shared features, right?
Absolutely. Whether it's a prokaryote or eukaryote, every cell has three main things: a plasma membrane, which acts like a barrier keeping the insides in and the world out, cytosol, the jelly like fluid filling the space, and ribosomes, which are like 3D printers making proteins. Oh, and of course, chromosomes. Can't forget about DNA.
Sounds like the cell has its essentials, no matter how complex or simple things get. And I know we're heading into even cooler territory next, with how everything works together.
Oh, we totally are the endomembrane system, my friend. This is where we really see teamwork come to life in a cell. Can't wait to dive into that next.
Exactly as we hinted at earlier, Eric, the endomembrane system really brings that teamwork concept to life. It's such a fascinating process where every part knows its job and works in perfect sync. It's honestly one of the best examples of collaboration you'll find inside a cell.
All right, you've mentioned it like it's a big deal. Let's unpack this. What's the endomembrane system all about?
Okay, imagine a giant assembly line. Think about how a car factory works. You've got different stations, each with a specific job, and they pass the pieces along until the final product rolls out. The endomembrane system is exactly that, but inside a cell. It's a network of organelles that work together to make, process, and ship proteins, among other things.
All right, I'm tracking. What's the starting point of this whole assembly line?
It all starts with the nucleus, which, remember, is like the control center of the cell. It houses all the DNA, the cell's blueprints. When it needs to send out an order, it photocopies its instructions into RNA, the digital copy, if you will, and sends that information out through tiny little nuclear pores to where ribosomes are waiting to get to work.
So the ribosomes are like the workers building the protein based on those instructions?
Exactly. And those ribosomes happen to sit on the rough endoplasmic reticulum, or rough ER, which is the first stop in making proteins. It's like the initial assembly station. From there, the proteins are packaged into vesicles, think of those as little shipping boxes, and they get sent to the Golgi apparatus for finishing touches.
The Golgi. So that's like the quality control or the packaging department?
Yep. The Golgi apparatus fine-tunes the proteins, maybe adding a sugar here or there, like a shipping label, so they know exactly where to go. Then another vesicle packages up the final product so it can be sent to its destination, whether that's inside the cell or out into the body, like antibodies attacking bacteria.
That's amazing. It's so intentional and streamlined. But not all cells are identical. Can we pivot to how plant and animal cells compare here?
Absolutely. So both plant and animal cells have these organelles working hard, but plant cells have some pretty unique features too. For instance, they've got a rigid cell wall, which gives plants their structure. Imagine how trees stand tall. And speaking of unique, plant cells have this huge central vacuole, which stores water, nutrients, and even waste. It's like their personal storage unit.
And what about animal cells? Does the lack of a cell wall give them more flexibility?
Exactly, exactly. Animal cells can change shape and move more freely because they don't have that rigid layer, and they rely on this dynamic interaction between their organelles to stay busy, producing energy, making proteins, breaking down waste. But it's that flexibility that's crucial for cells like muscle or immune cells.
So, whether you're a plant or animal cell, it seems like every part knows its role. Honestly, this is mind blowing. Any final thoughts, Dr. Rosario?
Yeah, I think what's really remarkable is how all these parts work harmoniously together, right? A cell's complexity is what enables life to function at every level. I mean, the studying and any of this just reminds me how interconnected everything is in biology.
And it's the perfect example of how science can teach us about efficient systems. Well, that's all the time we've got for today. Dr. Rosario, as always, your passion and insights are inspiring.
Thank you, Eric. And thanks to our listeners for tuning in. Until next time, keep exploring the world of cells. They've got more to tell us than we might think.
And on that note, we'll see you next time. Take care, everyone.